Hello students, welcome to the class of biochemistry. Myself Dr. Surekha, lecturer, Department of Biochemistry, SRM Dental College, Ramapuram. Today we're going to learn the process of replication of DNA. Now we'll move into the lecture. Before moving into the topic replication of DNA, we'll see the introduction. We all know DNA is the store of genetic information. It also gets to store the biological information of the cell. So during the cell multiplication, the biological information flows from DNA to RNA and to proteins. And this process is called central dogma of life. So during your multiplication of cell, the DNA gets multiplied. The process of DNA multiplication is called replication. And the replicated DNA gets converted into RNA. So the process of conversion of DNA to RNA is called transcription, and the transcribed RNA gets converted into protein. So the process of conversion of RNA to protein is called translation. So central dogma of life includes the replication, transcription and the translation to form a daughter cells. And also the function of each cell is controlled by the information stored in the DNA through the protein synthesis. So the function of each cell is determined by the DNA's information through its protein synthesis. Here you can see the picture of central dogma of life. And we already know that DNA is a genetic information store, so it is a major store of genetic information. And when they ask for the replication definition, you have to write this. Like during the cell reproduction or cell multiplication, the genetic information transfers from the parent cell to the daughter cell by the process of duplication or replication. So duplication of the or synthesis of DNA is called replication. And replication always happens in a semi-conservative mechanism, which means the strands will be consisting of one parent strand and one newly synthesized strand. Here the picture showing how the parent strand gets unwind first. The two strands get, the two strands get separated, and each strand can synthesize its daughter strand in order to form a semi-conservative daughter DNA molecules. In this slide, you will be seeing the replication of DNA in prokaryotes. Because the replication of DNA in prokaryotes is very well understood when compared to the replication of DNA in eukaryotes. Actually, this lecture I will be covering more of replication of DNA in prokaryotes. And in the end of the lecture, I have given the important enzyme involved in the replication of eukaryotes. And we'll go into the lecture first. So I told you already, the replication of DNA in prokaryotes is very well understood when compared to eukaryotic DNA replication. And the enzyme involved in the prokaryotic replication is DNA polymerase. It is a very important enzyme involved in the replication of prokaryotes, and it requires DNA template because the replication takes place as a semi-conservative mechanism. I already told you, like a semi-conservative mechanism means it uses the parent strand as a template. So it uses strands of one of the two strands of parent strand, and it uses as a template to synthesize the new daughter strand, so that the daughter molecules or daughter DNA molecules will be formed. And the DNA polymerase also essential for other important functions such as DNA chain elongation, DNA repair. So if there is any repair occurs in the DNA, the DNA polymerase only comes into play and it repairs the DNA. And the repairing action can also be mentioned as 5' to 3' exonuclease activity, and it also involved in the proofreading. Proofreading is nothing but any mismatch in the nucleotide can be avoided by doing the proofreading. So mismatch in the base pair during the replication or during the repair mechanism, so any mismatch in the nucleotide occurs. So in that that will be avoided by the DNA polymerase because it has got proofreading activity. And the proofreading is also denoted as 3' to 5' exonuclease activity. I already explained you the DNA polymerase is an important enzyme involved in the replication of DNA in prokaryotes.
There are three different types of polymerases is present in the prokaryotes. So DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. These are three different types of polymerases present in the prokaryotes. So DNA polymerase 1 involves in the chain synthesis between the Okazaki fragments on the lagging strand. So I'll explain you what is Okazaki fragments, what is leading, what is lagging stand in the upcoming slide. I'll tell you what is the important essential function performed by the different types of polymerases in the prokaryotes first. So DNA polymerase 1 involves in the synthesis, it just involves in the completion of the chain synthesis between the Okazaki fragments on the lagging stand, and DNA polymerase 2 involved in the proofreading and DNA repair mechanism, and DNA polymerase 3 involved in the leading and the lagging stand synthesis. Unlike DNA polymerases in prokaryotes, the eukaryotic cell consists of 5 DNA polymerases. They exist as alpha, beta, gamma, delta, and epsilon. The functions of the DNA polymerases of eukaryotes has been given in the last slides. So I'll explain that later. Now we have come to stages of replication. So the replication is a process which involves three important stages, that is initiation, elongation, and termination. So the replication of DNA involves three important stages, and I'll tell you in this lecture what will be happening during the stage of initiation and during the stage of elongation and during the stage of termination. So each DNA has to undergo initiation, elongation, termination in order to synthesize the new daughter strand of DNA molecule, so in the process of replication. Now we have come to the first stage of DNA replication, that is initiation. So during the initiation, the complementary strands of DNA get separated or unwind, resulting in the formation of the replication fork. So in the picture you could see the double-stranded DNA gets separated, resulting in the formation of the replication fork or replication bubble, and followed by the double-stranded twinned structure. So the replication fork is like a bubble formation in between the double stranded DNA. So this bubble formation occurs at the particular site. I mean, they occurs at a specific site or at the particular DNA sequences. That site is called origin of replication. So in the circular DNA, because the prokaryotes will be consisting of the circular DNA. So in the circular DNA, it will be consisting the particular DNA sequences at the specific site. That is called the origin of replication, where the replication fork occurs. And during the replication process, the DNA synthesized in both the ways, so they are said to be bidirectional. It is denoted as a red color arrow marks in the picture. The various steps involved in the initiation process is given here. This involves different proteins and enzymes. The first is the DNA-A, which is a protein, binds to the ORI region. ORI is nothing but origin of replication region, and successfully denatures the DNA. After the DNA denaturation, the DNA B, that is a helicase, DNA helicase protein, binds towards and unbinds the parental DNA using energy as in the form of ATP. So that the helicase enzyme move in both direction in order to form a proper unwinding, and resulting in the formation of replication bubble and two replication fork. During the unbinding process, there will be a generation of stress in the double helical structure. This is prevented by the topoisomerase by cutting either one of or both the strands of the DNA. And the unbinded protein tend to reassociate or tend to recoil, which is inhibited by the SSB, that is single stranded binding protein. So the single stranded binding protein goes and binds to the strands of the DNA, stabilizes it, and thereby preventing its reassociation or recoiling. In order to initiate the synthesis of DNA, we require DNA polymerase 3. DNA polymerase 3. The DNA polymerase 3 in turn requires RNA primer. The RNA primer is a short piece of the RNA which is of 5 to 50 nucleotides in length, which is synthesized by the enzyme called primase. Primase is otherwise called as RNA polymerase. So the RNA polymerase or primase synthesize this RNA primers using DNA as a template. So by using DNA as a template, the RNA primase synthesizes the RNA primers, so that once the RNA primers get synthesized, the DNA polymerase 3 can add the base pairs to the RNA primers in order to elongate the synthesis of the DNA. This is how the DNA gets elongated or replicated. In this slide, you can see the initiation stage of replication. I have given two pictures. The first picture denoting the replication fork, showing the origin of replication, helicase, single stranded binding proteins, primase, etc. So see here, the second picture showing the detailed initiation stage of replication. The first is the helicase enzyme, which involves in the unzipping of the complementary strand. So here the helicase enzyme separates the two strands by unzipping it and resulting in the unbinding of the complementary strands of DNA. The strands are stabilized by the single stranded binding proteins, and the stress developed in the DNA strands is reduced by the topoisomerase. The topoisomerase is also called as gyrase, and the RNA primers is synthesized from the DNA primase. Primase is an enzyme involved in the synthesis of RNA primers using DNA as a template. So using the parent DNA as a template, the primer will be synthesized from the primer will be synthesized by the enzyme called primase. So once the RNA primers get synthesized, the DNA polymerase starts adding the base pair to the RNA primers, thereby elongating the structure of the DNA. So the DNA polymerase present in the prokaryotes, they do not synthesize the strand on its own. They require RNA primer in order to initiate the synthesis of the replication. So the prokaryotes require RNA primer in order to initiate the synthesis. Thereby the primase enzyme synthesizes the RNA primer. This in turn helps the DNA polymerase to elongate the strands of the DNA. In this slide, you will be seeing the replication bubble. I have given same points as before. The only thing I wanted to emphasize here is the replication bubble is a very important short note question which can be asked separately also, the replication fork, replication bubble. So you need to draw a picture and you can have to explain this. And see, the replication bubble, I already told you, it is formed at the site of the replication by unwinding the two strands of the DNA. So the two DNA strands, they unwind at the site of replication to form a bubble. In prokaryotes, the eukaryotes, it has multiple origin of replication, so multiple replication bubbles will be formed in the eukaryotes. So in the replication bubble, the DNA synthesis occurs in both the directions, so it is called bidirectional and at the point of origin. So here the picture showing the strand of DNA, the template strand and the daughter strand, formation of a replication bubble and resulting in the separation of two parent and daughter strand molecules. Now we have come to replication fork. This is the same as replication bubble. I have already told you the complementary strand, they unwind at the site of origin of replication to form a replication fork, and it is the place where active synthesis of DNA occurs, and it do consist of 4 different enzymes of proteins such as DNA helicase, SSB, primase, DNA polymerase. So primase, helicase helps in unwinding the DNA, and SSB helps in preventing the recoiling, and primase helps in synthesizing the short fragment of RNA, and DNA polymerase helps in synthesizing the new strand of DNA. So this we have already told you, and also here the picture showing the replication with replication bubble, it forms first and followed by replication fork. So this replication fork can be asked in your short notes, so you have to write the notes with the picture. So I wanted to emphasize more on this. Now we have come to the second stage, that is elongation. So after the formation of replication fork, the DNA polymerase 3 started the initiation of the synthesis of new strand by adding the deoxynucleotides to the 3' end of the RNA primer. Thereby both the strands in the DNA are synthesized simultaneously in the opposite direction, one towards the replication fork and other away from the replication fork. Now we have come to the leading strand and the lagging strand. These two strands are synthesized during the elongation stage of the replication. So one of the strand of the DNA template produces the leading strand. The other DNA template produces the lagging strand. So in the leading strand, the DNA run from 3' to 5' end, and it is copied by DNA polymerase 3 as a continuous strand. So the nucleotides will be added continuously to one primer to produce the leading strand. Whereas the lagging strand, they run from 5' to 3' end, and it is copied by the enzyme DNA polymerase 3 as a discontinuous strand. So which requires numerous RNA primers. So the extended RNA primers with the nucleotide bases by the DNA polymerase are called short pieces of DNA, which is referred to as Okazaki fragments. So the lagging strands will be consisting of Okazaki fragments, whereas the leading strand do not contain Okazaki fragments. Okazaki fragments are nothing but the short pieces of DNA which is produced using the RNA primers, where the DNA polymerase 3 adds the nucleotides to it. These short fragments or short pieces of DNA are called Okazaki fragments. Now we have come to the slide showing the picture of elongation stage of replication. See, before going to the elongation step, there is a initiation step which involves the enzyme helicase topoisomerase SSP. So helicase is the enzyme involved in unzipping the complementary strand. During the unzipping process, the steps is developed. This is reduced by topoisomerase. The parent DNA strands are stabilized by the protein called SSB. Here they have also mentioned the direction of movement of replication fork. So the DNA parent DNA strands, they are represented in green color. One is from 3' to 5' and other is from 5' to 3' end. So the 3' to 5' end parent strand act as a template for synthesizing the lagging strand, whereas the 5' to 3' end, they act as a template for synthesizing the leading strand. Since 5' to 3' end, they act as a template for the leading strand. So the DNA synthesis, it run from 3' to 5' end. See, it requires only one RNA primers, and DNA polymerase 3 keeps on adding the deoxynucleotides to the RNA primers, resulting in the continuous strand which is referred to as leading strand. They run from 3' to 5' end. Whereas which is also found to be towards the replication fork. So the synthesis of DNA of leading strand is towards the replication fork, whereas in the lagging strand, it takes 3' to 5' as a template. So it runs from 5' to 3' direction, and it requires numerous RNA primers. To the RNA primers, the DNA polymerase adds the nucleotides, resulting in the formation of the short fragments of DNA, or referred to as Okazaki fragments. They are found to be discontinuous, and after the completion of the lagging strand, the RNA primers has to be removed, because it is completely DNA. So RNA primers has to be removed, which is done by the enzyme called DNA polymerase 1. So you have to be very careful that DNA polymerase 1 involved in the removal of the RNA primers, whereas the DNA polymerase 3 involved in the addition of the deoxynucleotides to the RNA primers. So once the RNA primers gets removed, there will be a formation of the nick. This nick is sealed by the enzyme called DNA ligase, catalyzes the formation of phosphodiester bond to seal the Okazaki fragments, resulting in the complete formation of the strand, completion of the lagging strand. So this complete lagging strand formation, they run opposite to the replication fork. We now come to the last and the final stage of replication, that is termination. The termination involves the specific sequences such as termination sequences, which is also called as TER. So the termination sequences is recognized by the specific protein called TER binding protein. So when the helicase unwinds the DNA, the termination sequences gets exposed. So to the exposed termination sequences, the TER binding protein comes and binds to it, thereby preventing the further unwinding of the DNA, which result in the termination of the replication. So like once the termination sequence is exposed to the TER binding protein, it prevents the helicase enzyme from further unwinding of the DNA, thereby facilitating the termination or stop of the replication. Now we have come to an important topic, that is proofreading. The proofreading is considered to be an important aspect in case of replication. So the DNA polymerase with high fidelity, it copies the parent DNA with high accuracy. Sometimes incorrect nucleotides gets incorporated during the replication process in the frequency of 1 in 108 to 1012 bases. This could lead to the mutation. Sometimes the mutation may be deadly. So the error ratio during the replication is kept low due to the specific process performed by the DNA polymerase, that is proofreading. All the three DNA polymerases have 3' to 5' exonuclease activity, that is otherwise called as proofreading activity. The DNA polymerase 1 and 2 are known to exercise the mismatched nucleotides before the introduction of the next nucleotide. So the all the DNA polymerases can perform the proofreading activity, among which DNA polymerase 1 and 2 can exercise the mismatched nucleotide, which can remove the mismatched nucleotide before it introduces the next nucleotide, so that the mutation can be avoided. So the proofreading is one of the important aspect. It has to be kept in the mind during the replication, so DNA polymerase can able to do the proofreading in order to accurately copy the parent DNA. Now we have come to eukaryotic replication of DNA. Previously, we have seen the replication of DNA in prokaryotes and proofreading. And now we will see the eukaryotic replication. This is closely resemble that of prokaryotes. However, there are certain differences exist in the eukaryotic replication, which includes it has a multiple origin of replication site. In unlike prokaryotes, some prokaryotes consist of only one origin of replication in the circular DNA, whereas in eukaryotic replication it consists of multiple origin of replication, and it also got 5 distinct DNA polymerases, which is which exist as alpha, beta, gamma, delta, and epsilon. So the function of DNA polymerase alpha is responsible for the synthesis of RNA primer for both the leading and the lagging strands of DNA. The DNA polymerase beta is involved in the repair of the DNA. Its function is comparable with DNA polymerase 1 found in the prokaryotes. The remaining functions of three polymerases is given in the next slide. So you have to remember the functions of each polymerases in the eukaryotic replication of DNA. Here the DNA polymerase gamma participates in the replication of mitochondrial DNA, where DNA polymerase delta is responsible for the replication on the leading strand of DNA, and it also got proofreading activity. And the final DNA polymerase epsilon is involved in DNA synthesis on the lagging strand and proofreading function. So these are all the various functions of DNA polymerases. Now we have come to an important topic, that is inhibitors of replication of DNA. The DNA replication can be inhibited by various chemicals or antibiotics. Mostly the inhibitors can directly target the enzymes, thereby inhibit the replication of the DNA. Mostly the enzyme targeted is topoisomerase or gyrase in the prokaryotes. So the prokaryotic topoisomerase or gyrase is inhibited by antibiotics such as ceprofloxacin, novobiocin, nalidic acid. These are the different antibiotics which can inhibit the topoisomerase activity, and they can be considered as an antibacterial agent. They can directly block the replication of DNA, thereby inhibiting the cell multiplication. But this chemical, they don't have any role on the eukaryotic enzymes. The human topoisomerase cannot be inhibited by the action of this chemicals. However, there are certain chemicals that act on the human topoisomerase enzyme and inhibits its function. Therefore, they are called as anti-cancer agents, which includes adriamycin, etoposide, doxorubicin. These are all the chemicals that act as anti-cancer agents. Along with the chemicals, some of the nucleotide analogs that can also inhibit the DNA replication and can called as anti-cancer drugs, which includes 6-mercaptopurine, 5-fluorouracil. So these are all the nucleotide analogs that can be incorporated into the DNA, which don't have a complementary base pair, thereby the DNA replication can be inhibited. Thus, they are referred to as anti-cancer drugs. So with this, I'm winding up the lecture. We have seen in the previous slide the inhibitors of prokaryotic replication. In this slide, you have given the inhibitors of eukaryotic replication. So far we have seen the replication, what are the stages involved in the replication, which is initiation, elongation and termination, and proofreading. And I have given the eukaryotic replication, the enzymes involved in the eukaryotic replication, and also the inhibitors of prokaryotic replication and the eukaryotic replications. So if you have any other further clarification, please do mail me. Thank you. However, there are certain chemicals that act on the human topoisomerase enzyme and inhibits its function. Therefore, they are called as anti-cancer agents, which includes adriamycin, etoposide, doxorubicin. These are all the chemicals that act as anti-cancer agents. Along with the chemicals, some of the nucleotide analogs that can also inhibit the DNA replication and can called as anti-cancer drugs, which includes 6-mercaptopurine, 5-fluorouracil. So these are all the nucleotide analogs that can be incorporated into the DNA, which don't have a complementary base pair. Thereby, the DNA replication can be inhibited. Thus, they are referred to as anti-cancer drugs. So with this, I'm winding up the lecture. We have seen in the previous slide the inhibitors of prokaryotic replication. In this slide, you have given the inhibitors of eukaryotic replication. So far we have seen the replication, what are the stages involved in the replication, which is initiation, elongation and termination, and proofreading. And I have given the eukaryotic replication, the enzymes involved in the eukaryotic replication, and also the inhibitors of prokaryotic replication and the eukaryotic replications. So if you have any other further clarification, please do mail me. Thank you.